Greetings everyone, a very warm welcome to sqadia.com. Paleness, dizziness, irregular heartbeat, weakness, chest pain, hemoglobinuria, which means hemoglobin in urine, and dyspnea. All of them predict a problematic blood or RBC function. This thread of signs and symptoms is heading us to think about RBC rupture, which is commonly known as hemolysis. In this video, we will go through different aspects of hemolytic anemia. Hemo means hemoglobin, which is a protein in RBC or red blood cell. Broadly, we can say hemo depicts RBC and lysis means destruction. Hence, hemolysis means breakdown of RBCs. Too much hemolysis can lead to hemolytic anemia. And let's see what is anemia. Anemia means decrease in amount of RBC to meet the body needs. Therefore, hemolytic anemia can be defined as type of anemia due to reduction in lifespan of RBCs, which is normally 4 months or 120 days. RBCs have a protein called hemoglobin, abbreviated as HB. It consists of alpha chain, beta chain, and heme group. Heme group has iron at its center. Iron is a mineral that the body needs for growth and development, which binds oxygen molecule. Each hemoglobin molecule has 4 heme groups, which can bind 4 oxygen molecules. RBCs don't have nucleus and possess a smaller number of organelles to accommodate more hemoglobin. Breakdown and recycling of RBC components is on the shoulders of liver. Hemolysis of RBCs start with Kupffer cells, which are specialized phagocytic agents of liver and break down hemoglobin into heme group and globin. Various enzymes such as peptidases convert globin into amino acids. On the other hand, heme group is broken down into iron and bilirubin. Iron is either stored in liver via ferritin protein or transported into the bone marrow via transferrin protein, which is used again in new RBC synthesis, while bilirubin is excreted in bile. One of the most common lab findings which predict hemolysis include elevated levels of unconjugated plasma bilirubin, which is a breakdown product of heme. Second most common finding is raised LDL or lactate dehydrogenase plasma levels coming from damaged RBCs. Another finding is lower levels or absence of plasma haptoglobin. Let's see what is haptoglobin. Haptoglobin is a protein made by liver which catches free plasma hemoglobin. In case of larger RBC destruction, haptoglobins will be insufficient temporarily because haptoglobin utilization is faster than the liver can produce it. Blood analysis reveals polychromasia. Poly means many and chromasia means color. It occurs when RBCs show up gray or blue upon dry treatment, indicating high levels of RNA, which depicts immaturity of cells as they are released too early from bone marrow than the normal red blood cells. Raised reticulocyte count is another feature of hemolysis. Both polychromasia and raised reticulocytes indicate increased red blood cell production. In case of cardiac hemolysis, which is kind of intravascular hemolysis, free plasma hemoglobins are released, which are filtered by glomerulus, followed by alteration in proximal tubules of nephron, where hemoglobin iron is converted into hemosiderin. When these tubular cells shed later into urine, it causes hemosideruria, which imparts brown color to urine. On failure of hemoglobin absorption by kidneys, it results in hemoglobinuria, which means hemoglobin in urine. In the next part, we will study causes of hemolytic anemia, which revolve around congenital and acquired categories. So keep watching sqadia.com.
From medical students to seasoned professionals, sqadia.com offers engaging and dynamic content for your educational journey.